So now let's synthesize some of that DNA. There's a lot of it to synthesize. Remember, this is occurring in the nucleus of eukaryotic cells where the chromatin exists, and it is occurring during the S phase of interphase of the cell cycle. So what is the synthesis process? So the DNA synthesis. What enzymes do it and who's involved? I'm gonna take a little sidebar and say a polymerase is a type of enzyme that synthesizes nucleic acid strands. So there are a lot of different polymerases, but when you hear the term polymerase, what you're talking about is the synthesis of a new nucleic acid strand. So that can be a DNA strand or an RNA strand, and that is a polymerase reaction. So we're going to be talking about polymerases are going to be the enzymes that are going to do the synthesis process. So the DNA polymerase is going to be the major enzyme. It's going to do the bulk of the synthesis, but there's a problem with DNA polymerase. It cannot start from scratch. Okay, so here's our single-stranded DNA parent strand. DNA polymerase cannot start from scratch, so you have to use a specialized enzyme called primase. So primase gets things started. It is a special type of, it's a type of RNA polymerase. And polymerase, shorthand, cool scientists call it Pol. So an RNA polymerase. And it adds a short RNA primer to get the synthesis started. And the primer is short, and what I mean by short, we're talking about usually less than 10 nucleotides in length, and it's complementary to the parent strand. And it is also going to be synthesized in a 5' to 3' direction. So depicted here in red would be a short RNA primer, and this would add, a handful of RNA nucleotides to get things started. Then the DNA polymerase comes in, and the DNA polymerase that does the bulk of the synthesis is DNA polymerase 3. Once this primer is formed, DNA polymerase 3 is going to add, it's going to extend the primer. It's going to add DNA nucleotides to synthesize the growing strand. And I want to take a look at the specific polymerase reaction that occurs. So here's sort of a cartoon. We've got our parent strand DNA in blue. Here's the base pairing. Should really be. These base pairs should really be dotted lines, dotted because they are hydrogen bonds. So what we see here is here's the parent strand, there's a C, the growing strand over here on the other, here's the 5' end, here's a 3' end. So it's growing in this direction. Always be able to recognize which end of the strand you're looking at. So here comes the last nucleotide in the strand is a G, and here's the structure. Here's the 3' OH. This is the magic. You have to visualize DNA polymerase in here, or whatever polymerase we're talking about. DNA polymerase in the active site recognizes this hydroxyl group in the incoming nucleotide deoxy nucleotide triphosphate. So the substrate here is the 3' end of the growing strand in a nucleotide triphosphate, 1, 2, 3. Well, wait a second. I thought, don't we only have one phosphate in the strand? And no, we ultimately in the strand will only have one phosphate, But what happens is this oxygen, the electrons on this oxygen attack this phosphorus and form a covalent bond. And these two phosphates leave the molecule. This is called pyrophosphate. This is a super high energy molecule, and it can be hydrolyzed and broken down to generate some additional energy, which helps to power this reaction. So DNA polymerase catalyzes the synthesis of a phosphodiester bond between the 3' OH and what's called the alpha phosphate of a nucleotide triphosphate. The beta and the gamma phosphate, so this is alpha, beta, and the gamma phosphate. The beta and the gamma phosphate leave as the molecule called pyrophosphate. And what you end up with is a strand that has one additional nucleotide in this new phosphodiester bond. So that's the chemistry that occurs. And I'm going to try to kind of write it in just a formula for what happens. So the DNA polymerase reaction, in the reaction, what we have is we have a strand, a growing strand, or newly synthesized strand, the 3' OH on a growing strand binds to a deoxynucleotide triphosphate. So it could be ATP, DATP, GTP, etc. And so if we think about this, we've got our strand with N nucleotides in it. In comes the deoxynucleotide triphosphate. The enzyme polymerase, DNA polymerase, is going to catalyze the connection of this hydroxyl to one of the phosphates here. Pyrophosphate, PPI, is going to be removed, and we have remaining this new strand with an additional nucleotide or with an additional nucleotide. Let's take a look at this from the perspective of a replication bubble. Remember, this is going to be opening in both directions, so replication is occurring in both directions. Let's put some directionality on here. We'll put 5' here, we'll put 5' there. We're going to have primase come in, and let's start with our, let's look at only this half of the fork right now. So we're going to have our primer come in. We'll make it in blue, and it's going to occur on both strands. But which way is it going to occur? Let me get rid of this little line thing here. It's going to come in. Primase comes in, makes a strand. Now, if this is the 5' end of the parent strand, what is the 5' end of the primer here? The 5' end of the primer would be there. So 4, 5, 6, maybe 7 nucleotides are going to be added by primase, and then DNA polymerase is going to come in. And synthesis is going to continue towards the fork. This is called leading strand or continuous DNA synthesis. And it's continuous because it's going to continue until termination occurs. So leading strand synthesis is continuous synthesis, and it occurs towards the replication fork. Let's look at our top strand, however, and let's put a primer up here. Now, which way was the primer put in? If this is the 5' end of the parent strand, this is the 5' end of the primer. How are we going to synthesize this? We can't, DNA polymerase, no polymerase adds new nucleotides at the 5' end. So how are you going to synthesize this? Well, this has to be done in chunks, in segments. And so as the fork opens, a new primer is going to be added as the fork opens. So primase is going to play roles throughout the synthesis of this strand. And then DNA polymerase is going to come in and extend the primer until it reaches, extend the strand until it reaches the other primer. This is called lagging strand synthesis. Or discontinuous. In lagging strand synthesis, synthesis occurs in segments. So DNA is synthesized in segments. And there are some additional proteins and enzymes in play. So I'm going to erase this stuff here. And let's talk more about the lagging strand synthesis in the process. So it's synthesized in segments. Those segments are called Okazaki fragments. And what happens is as the fork opens, approximately, and it depends on the species, but let's say it's hundreds, in the hundreds range, about every 150 nucleotides. So as it opens up 150 nucleotides, a new primer, new RNA primer is added. DNA polymerase 3 comes in and then extends the primer. So synthesizes the new strand until the previous segment is reached. Until that next segment is reached. Now DNA polymerase 3, sorry, this is 3 in here. DNA polymerase 3, which made this little red line, is going to dissociate, meaning it's going to leave the DNA, and a different DNA polymerase is going to come in. DNA polymerase 1 is going to come in, and it's going to chop up those RNA nucleotides. It's going to remove and replace the primer. Because remember, the primer is RNA, and it's not sustainable to have RNA DNA, even though these base pair, they don't base pair quite the same as DNA nucleotides do. So it's going to bind to and replace the primer. What's it going to replace it with? It's going to replace the RNA primer with DNA nucleotides. So let's zoom into that process a little bit more. Let me kind of let's make this artificial. Here's DNA polymerase 1. It is going to chop that. It is going to cut the phosphodiester backbone of this RNA nucleotide. That RNA nucleotide is going to leave, and it's going to replace it with a DNA nucleotide. Do you know what enzymes that cut the phosphodiester backbone are called? The removal of the RNA is catalyzed by a specific activity in the polymerase 1 called a 5', because it's going in a 5' to 3', 5' to 3' exonuclease activity. So this is DNA polymerase 1.
It's a specialized polymerase, which has catalytic activity that both removes the primer using a 5' to 3' exonuclease activity and adds in new nucleotides with its 5' to 3' polymerase activity. So it's got a couple of enzymatic functions. It has a cutting function. It cuts the phosphodiester bond, but then it can also reform that phosphodiester bond using its polymerase activity. So we're not quite done. Yeah, we've got a mess up here, but we're not quite done. So the primer adds in stuff. Polymerase 3 does the major synthesis. DNA polymerase 1 removes and replaces the primer. And the last enzyme that comes into play for lagging strand synthesis is DNA ligase. Seals a gap between strands, because what's growing over here, we'll look at that in class, but there's going to be one gap that cannot be formed by polymerase. And it's going to seal that gap. It's going to create a phosphodiester backbone, phosphodiester bond between these strands. So DNA ligase seals or glues together, let's say seals the gap between the strands or the segments, right? So any little gap that's here is sealed by ligase. Now that's a different catalytic activity than polymerase activity, because what we would have here is there would be a phosphate right here and there would be a hydroxyl group right here. Remember that polymerase activity requires a triphosphate, and we don't have that in this molecule. So ligase is more like a glue, whereas polymerase is more of a synthesizing enzyme. Whew, that was a lot, right? Here's just an overview again. We've got our leading strand synthesis that goes towards the fork. Lagging strand goes away from the fork, but all synthesis goes in a 5' to 3' direction. And since there are two forks, the leading strand on one side of the fork is opposite of what's happening on the other side of the fork. And this is what we'll look at in class 1. So the synthesis continues on until termination. In termination of replication, it occurs when two newly synthesized strands meet. Well, how do new strands meet? Or when it falls off the end of a linear chromosome. So let's take a look at go all the way back to the beginning here. There we go. So here's our new strand being formed in red. It's occurring in both directions, bidirectional forks. When those new strands meet, when the new strands meet down here, DNA ligase is going to come in and seal them together, and then termination occurs. In linear, so these internal ones, replication is going to occur until the new strands meet. There's going to be one little gap in the backbone, and that's where ligase is going to come in and seal the gap. At the ends of the strands, the polymerase is going to keep going, and it's going to fall off the end of the linear strand on one of the strands. On the second strand, something special has to happen. So it's going to occur until it reaches the end of a linear strand. But there's a special process in linear chromosomes where one of the strands cannot be completely
cannot be synthesized. So this there's a special case in linear chromosomes where one of the parents or one strand is not completely synthesized. And we'll pick that up in the next lecture. So we're going to put a pause right here, and we're going to talk about this special case in the next lecture. But to end out this one, let's go ahead and watch a quick video animation to pull this all together.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs in a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.